Hello, everyone. This video is mainly to introduce the script we just completed. This Fiji script and its analysis functions. And a tutorial on how to use it. Alright, let's first take a look at the file name of the script. Vacuole counting and measure intensity. We have actually revised it several times. Now, this is version 1.1.1. So, what is it used to analyze? This is an image of confocal. These cells are infected by the fungus Candida. What we want to count is how many cells there are in total in this image, and how many Candida are present inside the cells. Because Candida actually appears inside the cells, it forms this vacuole-like structure. For these vacuole structures, we also want to quantify the intensity of the surrounding autophagosome marker, LC3. So, if you want to perform the analysis, let's first see what kind of images you need to prepare. This image consists of 4 channels. The first channel is the DAPI channel. It stains not only the DNA in the cell nuclei. If you adjust the image contrast, you will notice that when we increase the contrast, you can see that besides the nuclei, the Candida genome is also stained by DAPI. We use these small DNA aggregates to determine the fungal DNA, which is Candida DNA, and how many clusters there are. The second channel is actin. The actin channel helps locate and count cells. The third channel is Candida's cell wall staining. You can see it forms these ring-like structures. During quantification, these ring structures in the GFP channel are counted too. The fourth channel represents LC3, an autophagosomal marker. This is what we ultimately want to measure, the LC3 intensity around the vacuole. When setting up your images, remember the channel order. The first channel should be DAPI. The second channel is actin, the third channel is Candida GFP, and the fourth channel is LC3. With these channels, we can use this script to count cells using the actin channel. Then, using the DAPI channel along with a size filter. The smaller regions, namely those between 0.3 to 10 μm superscript 2, are considered to be fungal DNA. In the third channel, the Candida GFP channel, we count the ring-like structures to see how many there are. We also calculate the vacuoles by intersecting the fungal DNA and cell wall channels. Once we locate the vacuoles, we extend them 4 pixels outward and draw a circle around them. Next, overlay the LC3 channel on this circle. After extracting it, calculate the intensity of LC3. Then calculate the overall intensity of LC3. See how much vacuole can separate, thus obtaining the average intensity of LC3. Okay, now let's see how to use this program. What you need to do is put the data you want to quantify in the same folder. I put it in a folder named Input here. So now there are 2 files inside. Let me close this file first. Okay, next you need to is open this script and then click Run. Okay, a window will pop up, asking where the files are located. Just assign it to the location where your file is currently stored, then click Select. Okay, next it will automatically perform the analysis. This step is quite long, so there are many steps involved. But basically it will be fully automated. So you'll see it display the data being calculated in real time. Okay, after calculating these 2 images, you will see in the final table that the folder contains 2 file names. These 2 files have calculated the number of cells, the amount of fungal DNA, and the number of fungal cell wall, also the vacuole, and the overall intensity of LC3. And what its average intensity actually is. Okay, let's check it out first. That is, its analysis results. So in the previous folder, for example, opening file number 15, and let it run for a bit to show all the circles. After adjusting the image brightness, you can see the green is fine. And the intensity of LC3 can be raised a bit. We can minimize this window first. Alright, first, let's take a look. In the same parent folder as the input folder we just saw, you'll find another folder called Intermediate and Results Summary Files. Here you can see the summary table from before. It will be automatically saved in this folder. So when you open it, you'll see a file format readable by Excel. And this table is its content. So this is the content automatically saved into that folder. Okay, next let's look at a single image file. Each file actually goes through multiple intermediate steps. So we have saved some important information to let us check it. Now we see image file number 15. First, I want to know where the recognized cell positions are. This way we can find the cell locations of image 15. After dragging the zip file, you'll see it has marked out the location of each cell. Next, let's take a look at where its nucleus is located. This is where it marks the nuclei positions. Now let's take a closer look at the vacuole and its analysis results. Okay, these are the vacuole locations we found. After finding these vacuole locations, next, let's look at a file named extracted LC3 around the vacuole. If you open these files, you'll see inside the vacuole locations we just selected. After adjusting the contrast, you can see that it has extracted all the LC3 signals around the vacuole. We are actually calculating the intensity of this image, and then integrating it into this table. This is what the tutorial video is about.